clustered, regularly, interspaced, short, palindromic, repeats.
It's just a specific DNA sequence present in bacteria such as Streptococcus pyogenes and Escherichia coli strain, which provides adaptive immunity against bacteriophage viruses by degrading their genome. Adaptive immunity was believed to be a trait exclusive to eukaryotes. However, the discovery of CRISPR and Cas proteins challenged this notion by suggesting that many bacteria and archaea possess a complex adaptive immune system. As you can see, these are the cluster of short palindromic repeats. These repeats are regularly interspaced by another sequence called spacer. Upstream of CRISPR array there is Cas gene that produce Cas protein, a crucial component for double-strand break. Hence the name suggests CRISPR Cas. Only those bacteria possess this system, is capable to fight with virus. Most probably they acquire it through conjugation, a type of horizontal gene transfer.
This system works in three-step process as adaptive immunity in bacteria: adaptation, expression and maturation, and interference. First, adaptation.
When virus insert their genome into host cell first time, the REC-BCD exonuclease complex recognizes the exposed ends of the double-stranded DNA and initiates rapid unwinding and degradation of the DNA. This degradation continues until it reaches a specific 8-base pair sequence motif known as a Chi site. On specific note, Chi site is the key player in discriminating self and non-self DNA in Escherichia coli. They are present in high abundance within their genome. When a double-strand break occurs in their genome, the REC-BCD complex degrades only a short segment of self-DNA before its degradation activity is halted by the nearest Chi site. The Cas1, Cas2 complex acquires new spacers from the remnants produced during REC-BCD mediated DNA degradation, also known as protospacer. Then Cas1, Cas2 protein come into play, consisting of 2 Cas1 dimers and 1 Cas2 dimer, binds to the dsDNA protospacer. There is a protospacer adjacent motif, a PAM. This sequence is specifically recognized by Cas subunit, used to find protospacer. Cleaved intermediate serves as the substrate for integration of the spacer into the CRISPR array. During spacer acquisition, the 3-OH group of the protospacer performs a nucleophilic attack on the 5' end of the first repeat, initiating the process. The 3-OH group on the other strand of the protospacer then carries out a second nucleophilic attack on the 5' end of the complementary DNA strand of the repeat, which is located adjacent to the leader sequence. This reaction leads to the expansion of the CRISPR array, incorporating the new spacer and duplicating the repeat and repaired. The resulting product is an expanded CRISPR array that retains a genetic record or memory of the previous infection. This memory enables the host to recognize and prevent future invasions by the same pathogen.
Second, expression and maturation.
Infection by the same previous virus initiates the transcription of CRISPR in the leader region. The CRISPR array is transcribed into a precursor cRRNA that is initially long and contains all the memorized sequences of the invaders. This pre-cRRNA is then processed into mature guide cRRNAs. The guide cRRNAs consist of a spacer, which is a short segment of RNA that complements a sequence from a foreign genetic element, located at the 5 ends. The 3' end of the cRRNA contains a portion of the CRISPR repeat sequence. In type 2 CRISPR systems, the presence of a trans-activating CRISPR RNA is necessary for the processing of the pre-cRRNA. The anti-repeat sequence within the tracrRNA allows it to form an RNA duplex with each repeat sequence of the pre-cRRNA. This duplex is stabilized by the Cas9 protein. Throughout this maturation process, the cRRNA tracrRNA complex remains bound to the Cas9 protein. Ultimately, this processing leads to the production of mature small guide RNAs that are ready to guide the Cas9 protein for target DNA recognition and cleavage.
The third and last step, interference.
In type 2 CRISPR-Cas systems, interference requires the presence of a PAM sequence and perfect complementarity between the protospacer and the cRNA, particularly in the seed region located next to the PAM. The presence of a PAM sequence preventing the system from attacking its own CRISPR locus. Before cleavage, the hydrogen bonds of base pairs breaks, and sgRNA forms complementary with seed. After that, the HNH and RUVC nuclease domains of Cas cleave the complementary and non-complementary strands of the target DNA, respectively.
CRISPR-Cas9 application as a genome editing tool.
As we know, the scientists Emmanuelle Charpentier and Jennifer Doudna represented CRISPR-Cas9 system as an advanced gene editing tool. For that they won the 2020 Nobel Prize in Chemistry. Let's have their perspective for gene editing with two examples. First example, the Cas9 enzyme, guided by a specific RNA molecule, targets DNA and creates a break in both strands. This break can be repaired using a DNA template through a process called homology-directed repair, HDR. The second example, Cas9 combined with a deaminase enzyme. The modified Cas9 generates a single-strand break instead of a double-stranded break. The deaminase enzyme converts cytidine bases to uracil. During the repair process, the Nic is repaired, and the uracil is converted back to thymine, resulting in a change from a cytidine-guanine base pair to an adenine-thymine base pair.
Thanks and namaste. You may give a feedback in the down.